So we are going to be interested in doing promoter engineering. And something we can look at is how constitutive promoters have different strengths. And so here, from slides from Howard Salas, you can see some examples of different promoters, and you can get a proxy or a measurement of transcription rate, and we'll talk about how in just a couple of slides, in arbitrary units. And so you can see that the consensus, which sort of implies the strongest, most favorable promoter, most likely this is within E. coli, gives you this rate of 60,000 arbitrary units. And then when you make a mutation or change the minus 35 hexamer, you see this drop. And that's keeping the minus 10 region and everything else identical. So you go from 60,000 to 3,000. But then if you kept the minus 35 region the same and you change the minus 10 region, you see an even more substantial drop, suggesting that, really your minus 10 sequence is really important. And you can make changes in either of these regions, and you can try to move where their location is, but you can see that there's a space-dependent effect. Even when you have the strong hexamer regions, if you effectively try to make them not minus 35 and minus 10, which, as a refresher, is referring to the distance upstream of the site of transcription initiation. I hope I said that in an earlier lecture before the break, but in case I didn't. So when you have this arrangement, this is no longer really a minus 10 or minus 35, and therefore you've kind of broken the promoter. I mean, all of these still suggest that some transcription is happening, which may be the case, but those are weaker sequences. So we as engineers can take a few different approaches to this. We can rely on biologists to characterize different promoter strengths. They're actually not that interested in that historically. Historically, a biologist is more interested in understanding naturally occurring phenomena, these natural promoters that might be, behind the trip operon or something like that. So then we as engineers can do that characterization. And then if we're good as a community, we can sort of document these promoters as standardized parts, well characterized with some kind of annotation about our measurement, at least relative strengths, and we can then share that broadly with the community, either in a publication or online. And so one of these, so this kind of idea around biological engineering was one of the foundational principles in synthetic biology, and led a long time ago, around 2004, to the development both of the iGEM competition, which is the International Genetically Engineered Machines competition, as well as a registry of standard biological parts. And so this registry is both a physical registry and a digital registry. It's now been somewhat surpassed by the AdGene plasmid repository as in terms of a physical repository, where people might store parts and plasmids. But AdGene has very good documentation. It's just not oriented as much towards specific categories of parts. So, in the sort of mid 2000s, I think he was at the time a J. Kiesling Lab postdoc, eventual professor at UC Berkeley, Chris Anderson, was characterizing different promoters and other genetic parts. And so he created a collection that was effectively named after him, where there were just, individual mutations or sometimes in pairs performed in some of these hexamer regions. And then there were different measured strengths. And so then really since, maybe 2006 or 7 or something, anybody who wants to try multiple different promoters in E. coli can just, we can say, well, even though our system might be a little different, I'd expect that this promoter, the BBA J23114, which shows here a strength of 0.1, is probably about 3 or 4 times less strong than 107 here. and so forth. And so that's a way that, hey, you get your metabolic flux analysis prediction. It says you need to upregulate this promoter. You can just introduce a few choices. You could study and try to predict how strong is the promoter that it already has, if it's an endogenous gene, and then how strong do I need to make my promoter. How do they get these measured strengths? One thing that's really convenient about characterizing genetic parts associated with transcription or translation is that you can just couple them to a fluorescent protein. And so if you recall this diagram from, I think, our first homework assignment, all you need to do is vary the promoter region on, say, a construct, a certain cassette within a plasmid. You'd want it to be constitutive in this case, so that there's nothing else interfering, relatively speaking, with the production of this protein. You can make it a red fluorescent protein in this case. And this is the actual image from that site showing how promoter strengths were obtained. So these are normalized to the fluorescence that was observed in this case. And I think some other kind of normalization that's important here is usually to the amount of cells that you had, since you could just have more cells and end up with more fluorescence. But what you want to understand is how strong is the promoter for any given cell. So I'll pause for any questions here. Okay. So we've been talking about promoters. We're actually not going to spend too much more time on them. They are discussed a little bit in your homework, and in fact, the homework involves thinking about the actual calculations involved in trying to predict promoter strength. And so I like that problem. I hope all of you have some background with MATLAB or Python, or worst case with Excel to be able to deal with problem 3 from the homework. But you've got more choices than the promoter. And actually, there's been more attention given to the ribosome binding site. So let's talk about why. You've got promoters which are being, potentially affected by multiple things, meaning that your average inducible promoter has operator sites that are sometimes overlapping with your -35 and -10 site. And I think we'll go into that kind of anatomy related to transcription factors next time.